I'm Richard Hollingham, and this time in the Planet Earth podcast, unravelling the mysteries of our ancient ancestors and the wonders of deep-sea worms.
They've got like hair sort of thing, don't they?
Yes.
Yeah, you wouldn't really know.
Maybe a thin strand from a sweater or something.
Yeah,
like
exactly.
that.
Well, I hope they're not, because I just... Yeah.
This is rather like Night at the Museum. It's after dark, and I'm in the main gallery of the Zoological Museum in Cambridge. Towering above me is a giant sloth skeleton. There are cases of bones, stuffed animals, some sort of pterodactyl overhead. It's slightly spooky, and I'm here to discuss another extinct creature that we owe our very existence to. The first four-legged animals to move from the water onto the land. And with me, Jennifer Clack from the University of Cambridge and Stephanie Pearce from the Royal Veterinary College, who recently published new research on the appearance and movement of these animals. And we've got some of these fossils here, Jenny. Just describe them for me. Really just sections, almost sort of hand-shaped stones. But what are we looking at?
Well, we're looking at part of the head of an animal called Ichthyostega. Unfortunately, it's missing the front end. Here we've got some neck vertebrae, and then further back we've got its shoulders and the ribs of the thoracic region of the animal.
Now, looking at that, I can see really with the skeletons around us here in the museum, there's clearly these ribs here in you've got in your right hand, and then the other was really just a stone with these lines on it, which just about fits in the palm of your hand. Are these teeth here? Is this the head?
These are teeth, yes. This is the lower jaw here.
So this whole creature, I suppose, is what, cat size, small dog?
This one was about a metre long when we put it all together with its tail, but we have examples that are at least twice as the size of that.
So what, kind of metre, 2 metres, that sort of thing?
That's right, yeah.
And at the moment, you look at this, you, I suppose, figure out it's got ribs, it's got these fierce-looking teeth. But there were a lot of assumptions made about this creature, these first creatures to move out of the water onto land.
The first creatures to emerge from the water and start to evolve legs with fingers and toes, people assumed that these would have had legs with weight-bearing limbs and that they would have walked in the same way that modern four-limbed creatures walk, like a lizard perhaps or a salamander. And they envisaged these early tetrapods, as we call them, as giant salamander-like creatures. And that's how they were first reconstructed.
But there's no reason why they should have been that, other than we can look at animals today and think, oh, that's very similar.
That's right. I mean, that was the assumption, that what you see today is what was going on in the past. And as a first hypothesis, that's quite reasonable. But what we found is that that wasn't the case at all.
So, Stephanie, what did you do? What did you do to these fossils to find out what they were really like?
Well, over the last 3 or 4 years, what we've been doing is we've been looking at the fossils that are still embedded inside the rock, so fossils that we can't see on the outside. And to do this, we've been using modern technologies. We've been using micro-CT scanners. And this one specimen in particular, this one here called Mr. Magic.
Mr. Magic, what? Sorry, I've got to ask, why Mr. Magic?
This was something that my colleague came up with. He had young children and they were fond of the Mr. Men, so I think he just picked that one because it came to mind. And it is a very nice specimen.
Okay, so Mr. Magic. So you looked inside Mr. Magic here.
We looked inside Mr. Magic using micro-CT scan data, and when we looked at the vertebral column, the bones of the vertebrae that were still trapped inside the rock, we weren't able to identify all the different parts that were supposed to be there based on what had been described in the literature and in textbooks over about the last 100 years. And so this got us really, really puzzled. What was actually going on in the backbone of this animal? And what we decided to do was take it to the European Synchrotron Radiation Facility and create really high-resolution images using synchrotron X-rays. And what we found was something very, very interesting.
So you've got a representation of this, what you saw inside this fossil, on the computer screen here in front of us. And it's a three-dimensional representation. And this wasn't what you were expecting. I mean, there's clearly a backbone there. I mean, it clearly looks like anyone could see that that's some sort of backbone, but it's not quite what you were expecting.
It's not quite what we were expecting. When you talk about a modern tetrapod backbone, their vertebrae are composed of individual units that are all interconnected, and those units are composed of only one bone. An early tetrapod vertebral column is actually slightly different. It's actually composed of many different bones. And in particular, each unit is supposed to be composed of 3 separate bones. It's supposed to be composed of one bone in the front, which is called the intercentrum, and that's the green bone in the image here. One bone on the top, which is called the neural spine, and that's the pink bone in the image. And then a pair of little bones at the back called the pluricentra, which are the yellow bones in this image. But that sort of concept of what an early tetrapod vertebra looked like isn't actually what we found when we did the synchrotron scanning of this fossil.
So what did you find? What was different about it?
Well, some of the bones ended up being fused together. And in particular, the bone at the front, the intercentrum, was actually fused on to the back of the pluricentra. And this was very unusual because that indicated to us that the first bone in the series, the intercentrum, was actually, in fact, the last bone in the series. And this meant that all the literature and the textbook previously actually had the backbone back to front in these animals.
What does this actually tell you about the animal, what it looked like and maybe how it moved?
One of the things that we found, you can see represented by a series of blue elements. And the nearest equivalent we can find in modern animals is the sternum, the breastbone, which in most animals is actually a series of segmented bits rather than, as we have, just one bone. But there they were, and that corresponds to the idea that these creatures were not walking with alternating footprints as you would expect, but actually were using a kind of crutching motion. So using their front legs to kind of hop forwards and then pushing or stabilizing with the back legs, because the back legs were paddle-shaped. They weren't walking legs. They didn't have proper knees or ankles. They were stiff and probably were just sort of sitting there in the mud, used for swimming in the water, but as stabilizers and anchors on land.
So based on this study, what do you now know that they look like?
Well, one of the changes that has been made in our perceptions is that the proportions seem to be quite different from the idea of the inflated salamander that was the usual textbook or kiddie's book representation. Its shoulders were relatively enormous, and it had this big rib cage, which then faded out into a rather weak bit of backbone just before where the hind legs fit in. And that's why the specimens all fall apart round about there. And the hind legs were paddles set sort of vertically against the body and the little short stubby tail. So it's really an unusual looking animal. I suppose if you want to find a modern analogue for how it looked in terms of proportions would be an elephant seal, something like that. Big head, big shoulders, rather diminutive hindquarters.
And fearsome teeth.
And very fearsome teeth.
And Stephanie, this ties in with the previous work you've done on this and published on this about the way they move. So we're building up a whole picture about these creatures.
That's right. Finding the fact that we had a sternum in this animal was really exciting because the sternum provides support for the chest, which gave us further support for the fact that these animals might have been putting a lot of pressure on their chest. So as Jenny mentioned, if these animals are pulling themselves forward with their front limbs, they would have had their chest in contact with the ground during part of their locomotion cycle. And so having these bones in their chest would have allowed them to put their body weight on the chest and would have allowed them to have a rigid rib cage around them to support their body weight, which is really exciting. So this gives us some insight into the fact that these animals might have been making tentative steps onto land.
And that's the point, isn't it? Tentative steps, Jenny. These weren't land creatures.
The old idea was that you went straight from a sort of swimming action with fins, and then these finned creatures made forays onto land using their fins, and those gradually evolved into legs. And they used this sort of same lateral bending that you see in fishes, and as you also see in lizards, for example. But it turns out that they were probably exploiting many different kinds of locomotion at the beginning, and they weren't all equivalent to walking.
There was clearly, though, an advantage in evolutionary terms to being on land, and that ultimately led to amphibians, to reptiles, to us, I suppose.
Yes, that's right. I mean, it's probably a combination of things that led to the transition. Increased food in terms of the arthropod faunas, the centipedes and the spiders and things that they could eat. An increase in the amount of plant cover that would give them shelter. Possibly escape from predators who couldn't make it onto land. Big fish predators wouldn't have been able to follow them. Perhaps to bury their eggs. There are all sorts of things you can think of.
Jennifer Clack and Stephanie Pearce, thank you both very much. And you can see some pictures of these fierce-looking animals on Planet Earth online. You can also, of course, follow us on Facebook and Twitter. To understand the sheer variety of life in the seas, you have to study all of it, from the tiniest bacteria to the largest fish. And then there's the stuff in the middle, such as bristle or polychaete worms. Letitia Dunton studies these polychaete worms at the Natural History Museum's Darwin Centre in London. Now, the worms can reach up to 3 metres long, but as Sue Nelson discovered when Letitia brought out some of her collection, there are plenty at the other end of the scale as well.
They're quite small. They're actually in ethanol at the moment. So yeah,
Good
these are...
grief, they are small.
Yeah, they're pretty tiny. So they're about a couple of millimetres long. Yeah, the smallest ones are about, like 2 millimetres long.
You would never guess that they were actually worms looking at them with the naked eye, because some of them look like tiny...
They look a bit like hairs sort of thing, don't they? Yeah,
Yes.
you wouldn't really know.
Maybe a thin strand from a sweater or something.
Yeah,
like
exactly.
that.
Well, I hope they're not, because I just... Yeah. They're sort of like a brownish colour and a bit like reddy.
Yes, there's one small red one in there. Now, where did you get these worms from?
These worms are from the Whittard Canyon, which is an underwater canyon in the northeast Atlantic. So it's just southwest of Ireland.
And how big, large is this canyon?
It's pretty big. It's like, goes down, starts at a depth of 200 metres and goes down to 4,000 metres. These samples were actually collected from 3,500 metres. The canyon's about 2 kilometres long.
It's so funny that from such a huge structure you should be interested in
These
such tiny
little worms. Yeah, well, actually in the deep sea, most of the organisms are tiny because of the amount of food. There's not much food down there. So organisms have adapted to that by becoming very small.
So let's have a look at these tiny little polychaete worms under the
Put
microscope.
them under the microscope now. So I'll just turn this on. Lights come on. Just get this in focus. Right. Hope you'll be able to see these parts.
Oh, yes.
It's very hard to identify them sometimes because of the process of bringing them up. That obviously disturbs them a lot.
They're almost like thin white worms, but not so smooth all along in their body, slightly disjointed almost, or is that part of the process?
Some sort of segments. Well, actually polychaetes are related to earthworms that you find in your garden. Earthworms have little hairs on them for movement as well, and so do these polychaete worms. And they are quite small, sort of longish, they're all sort of this colour, but they weren't this colour to begin with, they were actually more vibrant colours. It's just the preserving process which makes them all this sort of...
Has sort of
whitish.
bleached them out a bit.
Yeah.
So why are you interested in these particular worms?
These particular worms. Well, polychaete's actually very abundant in the deep sea. I've been looking at macrofauna in general. So in the deep sea, when you're looking at deep sea biology, you actually divide animals, organisms up by sort of sizes. So you have the myofauna, which is everything very, very small. Then the macrofauna is a bit bigger. And then the megafauna is a lot bigger, so things like fish, the megafauna is. But I'm concentrating on the macrofauna. And macrofauna are things like polychaete worms, crustaceans such as isopods, amphipods, copepods as well.
So if there's so many of them, these must form I'm assuming must form a food source for other items in the sea.
Yeah, so probably get megafauna, so the slightly bigger animals grazing on them and everything.
And why these in particular?
Just because I'm looking at actually species diversity in the deep sea, so I want to understand why there's lots of species in a certain place. I'm working on submarine canyons, underwater canyons, and underwater canyons are thought to be hotspots for species diversity. And for me to sort of properly understand this, I'm looking at the polychaete worms in particular to find if there are lots of polychaete worms found in these underwater canyons. At the moment, yes, I found there are lots of polychaete worms in underwater canyons.
And are there lots of different species of these worms in particular? Is much known about them?
I think it's 12,000 species of polychaete worm
My goodness.
identified. Yeah, that's quite a few identified so far. So the ones that I'm looking at, they're probably, and to be honest, they're probably all unknown species, a lot of them, which makes it quite hard for me when I want to understand species diversity, if I'm finding lots of species that are unknown.
And when to my eye, they all looked very similar
Yeah.
under the microscope, particularly when
they do,
they've
don't
lost
they?
their
Yeah,
colour.
yeah.
How do you tell the different species apart?
With polychaetes, they obviously have the chaetae, so the chaetae are the little hairs going all along down the side of the worm. So you can look at the chaetae and also the prostomium, which is like the head end, so you can see the different shapes of that.
Now, one of the ways in which you must compare what you've got here with what you've got already in the Natural History Museum must be with the collections.
Yeah, so the collections are like a really fantastic resource I have here. I'm very lucky. I think there's 8,000 polychaetes in the collections.
Well, I think then we ought to go down
Yeah,
to
we
the
better. Yeah, let's go.
collections then.
Yeah.
Excellent. Feels as if we're going into an airlock.
Yeah, it's very high security here. Yeah. Well, this is to stop any contaminants coming through, like rodents or anything, yeah.
Right,
It's
past
quite cool
two
as well.
automatic doors.
Yeah, 2 automatic doors, and as you notice in here, it's actually a bit cooler.
Yes,
Yeah,
gosh,
it's
yes,
quite
quite considerably.
Outside, that's to help with the preservation of the specimens down here.
Now, it's filled with what look like giant filing cabinets.
Yeah, just masses and masses of giant filing cabinets. And if I actually open this one here, which says polychaete worms that I'm specialising on, open it up here. And we have all the type specimens and the voucher specimens. So
Lots of different shaped jars, glass jars here, all sorts of sizes. Some like the size of like a face cream, some that you'd have little herbs. You'd expect the size of herbs and
these
spices.
are sort of like Victorian.
They do.
And in fact, they probably are Victorian. Some of these are really, because they do actually have collections from Darwin and Cook so they're all stored here in the Natural History Museum.
Yes, that one looks very old-fashioned with its nice little glass
Yeah, it's
stopper
got a really
at the
nice
top as
little
well.
stopper and nice labels. Amazing handwriting. I wish my handwriting was that good. Yeah, so these are some of the Polychaeta.
How many worms are in that? Because they are so much bigger than the ones that we've looked at, which were the size of a hair, whereas these are more maggoty size.
Yeah,
a bit
exactly,
thinner.
yeah. So there should just be one in here. This is the type specimen. It's actually from a Challenger exhibition.
And
to
so you
compare
would then
them-
take this one, say, up to your lab,
Yeah.
look under it and compare with what you've actually got.
Exactly, yeah, exactly that, to see if it was the same species.
And what do you do when you have found a species that you can't find here in the Natural History
Yeah.
Museum collection?
Well, that's so in my deep sea biology ones, there are a lot of species that won't be here. So what you do is you describe it, so you have to like write down all the features, morphological, and now actually there's moving more to molecular. So you probably sequence it and then get the sequence information, the DNA from that, to like come up with a new species.
Letitia Dunton from inside one of the Natural History Museum's collection rooms on polychaete worms and the diversity of marine life. And that's the Planet Earth podcast from the Natural Environment Research Council. I'm Richard Hollingham, and from the Museum of Zoology in Cambridge, thanks for listening.